Hello again, everyone. My name is Steele Forrest. I'm the program manager for the Prepare Institute, which is essentially UC San Diego's Pandemic Preparedness and Response Institute, but we're actually more than that. Our acronym is so includes pandemic response to emerging pathogens, antibiotic resistance, and health equity. So it's if you haven't had a chance yet to look at our to learn more about us, please do take a look at our website. The URL is there in the chat for you. And then moving on, so today we're very pleased to have Dr. Ben Smarr presenting. He'll be speaking on the potential for augmenting public health through IoT. And Dr. Smarr is an assistant professor in the Su-Chen Jean-Lei Department of Bioengineering in the Halijodu Data Science Institute at UC San Diego. How was that, Ben? Was that passable?
That was like a 7 or an 8, absolutely.
Okay, great. I'll take it. That's great. All right, so a little bit more about Dr. Smarr. So Dr. Smarr's research focuses on time series analysis in biological systems. He emphasizes practical information extraction for translational applications. His lab develops novel approaches to get more information out of data generated over time and within individuals. His lab uses these approaches to increase precision in health algorithms, with a special focus on increasing equity of health tech efficacy over diverse populations. Dr. Smarr has received fellowships and funding from NSF, NIH, DoD, and many private partners. His background in biological rhythms and neuroendocrinology gives him a rare perspective into the current challenges for data science and engineering on biological systems. So with that, Ben, I'll go ahead and pause my share and you can take over. And just so everyone's aware, if you want to put questions in the chat, that's fine, but generally it's an open forum after the talk is done.
Great. Okay, I'm going to share a screen here if I can. And yeah, happy to take questions afterwards. You all are seeing my screen, hopefully not too much Zoom stuff, yeah?
Looks good.
Great. So you see that my hair comes and goes in length. I want to cover a fair amount of ground today. So I'm talking about this in terms of potential for augmenting public health through Internet of Things. Internet of Things means your cell phone, your wearable devices, your smart refrigerator, even your car. And the punchline is going to be, we all generate a lot of data as individuals right now, even without any extra effort. Things generate data around us anyway. Because we generate those data, they carry patterns that have to do with how our lives are going and when that's changing. And so I'm just going to try to show you a few examples, starting with a few more specific stories, building into COVID response, and then surfacing some of the challenges and the opportunities going forward. Challenges, mostly biases in data, equity of access, this sort of thing, and then expanding beyond the sort of on-person devices that I'll start with. So with that, let me just dive in. I work with a lot of groups on campus, so I want to highlight the Center for Circadian Biology, one of the great centers for biological rhythms research in the world, actually, and of course, my home departments. Let me see if I can hide this screen here. So as I say, I want to start with a story. This was done with Quantified Self, which is a community of people that are interested in putting numbers on effects in their lives that they don't have tools for capturing otherwise. A lot of health stories, a lot of other things as well. This was actually having a participant who was going to come join us for a conference at UCSD a few years ago. And they were flying in from the Netherlands. And I work in, among other fields, network physiology. And so what that means is, your heart rate's changing, your temperature's changing, your hormones are changing. They don't do this randomly. They do this with some coordination to each other if you're healthy. They all have to live in the same skin, so they all have to get to know each other. So here we said, hey, there's this rule in circadian biology that when you're jet lagged, your physiology gets messed up, you're going to fly in from the Netherlands. Can we put a couple of devices on you so that we can see if we can see that jet lag? And it said, sure. So we put on, in blue here, we have, let's see, I believe it's temperature. And then in green, we have glucose, continuous glucose monitor. And what you notice is that, let me get a little pointer. There's a daily rhythm. So, going up and down as they go to sleep. And then there's these little ripples with meals for sugar. But they look pretty lockstep, right? If you knew where green was bumping, you might anticipate where blue is bumping. If you knew where blue was going high, you might anticipate sugar is low, this person's asleep. And it repeats every 24 hours reasonably stably. We can mathematically quantify that level of coherence. And so what you're seeing here in white is what's called a wavelet coherence. We're going to skip the math today, but the idea is that around 24 hours, there's a very strong agreement. Backwards arrow means anti-phase, so opposite to each other. Very strong agreement between these two. So if you knew one, you could predict the other at the 24-hour phase, 24-hour rhythm level. Then they fly from the Netherlands and they get jet lagged. And sure enough, you can see that this no longer looks nice and orderly at the 24-hour rhythms. It's pretty jagged. The green glucose really just falls apart as a signal. And it takes them quite a while to come back to that pattern that they were at beforehand. Now we have 1 hump a day, 2 little glucose humps. Again, we put numbers to this. And what you can see is that in the literature, there's this idea it takes you about 1 day for every hour of jet lag that your body has to then resynchronize to. You're not just one clock. All your cells have these clocks. Trying to avoid all of the basic circadian stuff today, but suffice to say, your body has a lot of organizational challenges. It takes them that long to get back on track with each other. The point here is to say, we then see the reemergence of that synchrony statistically here in the hot band at 24 hours after about 7 or 8 days. And this is a 9-hour time shift. So that's just about what we would expect, right? This really was a verification of the literature, but it was more than that. It was the idea that if people were wearing these devices, generating data like this anyway, you could be doing, syndrome surveillance for this kind of jet lag desynchrony in an arbitrarily large population, right? You don't have to ask somebody, did you feel jet lagged? You can see it in their physiology. And so as we build systems that are capable of processing IoT data like this, that's really what the potential of the space is. People will generate these patterns. We can mathematically put numbers on them, and we can use that to then understand where are their physiological disruptions at great scale. Another example, I'll go through this fairly quickly. Individuals that were eating whenever they want to. So there's a raster plot of days and time of a meal, and they're reporting on their phone, when do they eat? And then a week when we tell them exactly when they're supposed to eat. And this is them signing in and saying that they're, in this case, being very compliant. Last day, there's a day of fasting, then they get to eat late at night. And again, we measure distal temperature at the wrist, an proxy for core temperature in the armpit, and continuous glucose with the Freestyle Libre. Again, mathematically, we developed little dashboards for these people. So the size of the dot is at the 24-hour level on the outside and at the what we call ultradian level on the inside. And this is really, you can think of this as meal rhythms, but it's a sort of 2 to 3 hour rhythm that occurs across hormonal and metabolic systems. The size of the dot is the scale of that rhythm. And then the fatness and the blueness of the arm is how coordinated were those rhythms. Again, it's that coherence that we just saw. And you can see that this person in general is not actually super coordinated. They eat sometime during the day, but there's not much structure to it. Then they get structured, and we can see again their physiology responding. They have these nice fat blue bands. All of a sudden, they seem very synchronized. And that ultradian power, which again, very important for endocrine regulation within a day rhythms, that gets much, much stronger here. And so again, there's a lot of talk you've probably heard about, time-restricted feeding or timed fasting or this kind of thing. The idea again is to say the subjective experience isn't the end all of that. The sensors are ubiquitous enough at this point that it's very cheap to get those data off of somebody living in the world and be able to monitor, is this kind of behavior change actually having a positive effect? In this case, the ambition being outpatient monitoring for things like metabolic disease. Then we looked at that day of fasting, and even though they're not eating any meals at all, so they're not sort of kicking the tires, nevertheless, the coherence across these systems at that ultradian scale stayed pretty high. Preliminary data, but it suggests that when you have people get their bodies into synchrony, their bodies do a pretty good job of staying synchronous, which is very good, gives us hope. So we took these tools, a bunch of related approaches, and we tried to apply them to COVID when that started. My whole lab at UCSD started in March of 2020, which was not a great time to be starting new research initiatives. But despite everything getting shut down, not getting lab space, because we had this kind of mathematical approach, we were able to work with some partners, Department of Defense and Oura Ring as a device provider, to be able to actually capture real life data from about 100,000 people that signed up. Um, that raised a lot of really interesting challenges for, okay, we can do this a few people at a time. Are there principles that let us do this at scale? So first big principle that I really want to make sure people take home with them is that people are different from each other. Not a shocker, but surprisingly absent from a lot of statistical analyses. So here we're looking at the first 50 COVID patients that we saw in this study. And we're just looking at on average, and these are overlaid, when they're healthy and when they're sick, what is their temperature range? So the Oura Ring is measuring temperature from the finger in this case, distal temperature. What you can see is that people are very different. They have some areas of overlap. They have some areas where they don't overlap. And if you imagine back to 2020, getting on a bus, somebody scans your forehead, that's looking for a single temperature, 100.4, above which you have a fever, below which you don't. But that's going to be very different for these people over here who are just naturally colder than it is for these people here who are just always warmer. So this idea that a single number accurately reflects the population, makes sense when that's the only tool you have. But having this abundance of data lets us get way past that very quickly, lets us have much more individualized metrics. So the next hurdle, aside from people being different from each other, is that people are different from themselves. We change very stably over time. This is illustrated here looking at temperature across 2 months. The arrow is highlighting where these individuals on average are having a fever as they become ill. And these big corduroy whales, these big up and downs, are their temperature changing between sleep temperature and wake temperature down here. So what you can see is, once again, if you just took the average temperature, it would be somewhere here in the middle. It would be completely useless, right? It would be basically the stopped watch equivalent of how accurate is your watch. Well, it's right twice a day. So using something like mean of aggregate data, we would never see this pattern. Instead, because we can see that everybody has this 24-hour rhythm to them, to their data, to their physiology, we can start to do things like, okay, well, let me look at just the sleep temperature or just the coldest of the wake temperatures and separate those extremes. And when we do that, what we see is a transformation, right? It goes from there's this tiny bleb, the mean would never reflect it in a million years, to highly significant change of just that maximum range. Here's the fever, very clear. And here's the minimum, right? Less clear, but nevertheless statistically significant, shakes out. You can even see it ramping up for several days. And in fact, this increase of daytime temperature, we've seen this in many cases now, and this seems to be generally a good indicator for inflammation in the system. So again, one universal temperature doesn't work across all people. One universal temperature doesn't work for any given individual, but contextualizing the phase of the time, in this case, time of day, for that individual, normalized temperature for that individual per time, makes this kind of aberration detection, here's your fever, go from completely impossible to actually pretty trivial. And this, again, this is a big hope that a lot more disease surveillance and syndromic surveillance is actually really at our fingertips. So we put this stuff together in pursuit of COVID detection systems. We ran a clinical trial in early 2021. When we were applying for funding, just for context, to remind everybody, the idea that wearables could help with disease was not obvious to most people. There's a lot of pushback on this idea. Whereas when you look at the data here aggregated across individuals with COVID in this study, you see these up and downs. These are daily rhythms again. We're looking at heart rate and heart rate variability on the top, daily and nightly temperature, those two things we just separated in the previous slide, here in the middle, and then respiration rate and MET, which you can think of as steps, sort of activity level, on the bottom. You don't need a degree in mathematics to see that there's a big change here. So here's the day that they got a test. What we can see is several days before they sought testing, their physiology was deviating. The heart rate was rising, heart rate variability was going down, temperature is rising, here's that fever. And then you can also see lasting effects, right? So right now, something that kind of surprises me is how little discussion long COVID gets. COVID has infected at least 1 or 200 million Americans, possibly more, certainly much more worldwide. And, it's killed probably or led to directly or indirectly tens of millions of deaths. But it also has something like a 20 or 30% rate of causing lasting change, lasting harm. You see this as soreness or shortness of breath, but you also see more extreme things. You have people that have persistent shortness of breath, or the damage happened in their pancreas and suddenly now they have diabetes, or it happened in their gonads and suddenly now they're sterile. 20 or 30% of hundreds of millions of people is a lot of people. And so there's going to be an enormous need for understanding who are these new chronic illness patients in our population, given that long COVID can impact us in very different ways depending on where the damage happens. We don't have sophisticated screening tools yet to understand who definitely has this syndrome. But that syndrome and others like it can start to be found in this kind of data. And I'm illustrating that just here in the bottom. For some individuals, they recover from their illness quickly. On average, what you notice is, for example, respiration rate stays elevated, sometimes for many weeks after infection. Similarly, activity stays low. They're not getting back to their normal daily routine for weeks, even just on average, right? So some individuals are faster, but some individuals are much slower. So having tools like this that allows us to say, okay, first of all, yes, we can definitely see when you're acutely ill. That's quite doable. That's actually fairly trivial. You might be able to imagine that this is put in service over populations. Hey, looks like a lot of heart rate and temperature rise is happening in Seattle today. Let's direct some resources there. I wonder if if there's an outbreak emerging. After that kind of acuteness, there's then the other side of it, where we could say, hey, of those people in Seattle that were part of that outbreak, which I'm making up, we can identify those individuals that have not returned to their individual baseline yet. Let's reach out and let's try to figure out what's going on. Can we do something preventive before that's debilitating for them? Again, we're not doing that currently. My point in this talk is to help raise the awareness that we could be doing that sort of thing. The technology is there. The distribution, the infrastructure, the equity, and the security are much bigger barriers. So getting away from disease for just 1 minute, we don't only want to know when things are bad, we also like to know when things are good. So the same approaches that we use here for COVID, we also use for women's health. Before I sort of let myself get drafted into service of COVID, a lot of my work was in women's health. Women's health is also extremely underserved. And so there was this issue of, could we use these wearable devices to tell when people were getting pregnant and monitor the health of those pregnancies? So I'll just show you a little bit of that here, but once again, very similar problems. Looking on average, even on normalized data, so people are normalized to themselves, so we don't have that across individual problem in this graph. Nevertheless, you can see that it looks pretty much like a flat line of temperature. Days is where most of the variance happens. And if you look very closely, you might notice that sleep temperature at the top sort of has these two humps and then stays high. And so if we zoom in on that, isolated as a feature, in fact, what we see is that across individuals, we can see menstrual cycles. So after you ovulate, your temperature and your heart rate rise. So we see that in the data. They then come back down, you menstruate, or sort of menstruate here, come back down, ovulate, go back up. And then here they ovulate and go back up. Having been impregnated, they get higher and they stay higher. So we can see this onset. On average, we can see that onset faster than your off-the-shelf pregnancy test lets you see it. And, for a lot of people that might not know that they needed to go buy a pregnancy test, again, this kind of passive biofeedback could be extremely helpful. We're then pursuing this further and looking at monitoring pregnancy outcomes, but I'm not going to get into that here. The story is similar to COVID. There are acute events that we can see. There are long-term events that have to do with management that we can also see. the challenge is not making this kind of thing real. It's real now. The challenge is getting it to be integrated in a useful way for syndromic surveillance and smart public health. So that brings us to what are some of these challenges? Here, what we're looking at is the ambitious work of a student, Patrick Castle, in my lab. And he said, okay, well, I want to look at fever detection as a syndrome, as a sort of a specific thing, instead of, is it the flu, is it COVID, is it pregnancy? Maybe we can focus just on, hey, your temperature went up in an aberrant way. So he was building some fairly sophisticated fever detection systems. Those work very well. And then he said, okay, cool. Now that I've done that, I want to come back and I want to look at everybody. Remember, we have tens of thousands of people across 2020 with all of their data. How good is this thing in sort of simulated in the wild? And so here, this big orange spike is fevers. Pardon me. What we're looking at in axes here is what's the relative contribution of these different physiological modalities, right? So as you're building a new sensor, a new smartwatch, or whatever it is, if you have a PPG sensor, photoplasmography, that allows you to see heart rate variability or heart rate, respiration rate comes out of that as well, which is just a modulation of heart rate, really. Those are all pretty much on the same line. What that's telling you is in this principal component space, so we've taken lots of information, we're projecting it into, in this case, just a 2D graph. All of that heart rate variability, everything is contributing roughly similar information. They're all together a single axis. And then here's temperature, which is a roughly orthogonal axis, so almost independent temperature from these heart-related outputs. So, that means that if you want to have better resolution on what's happening, it's really good that you have something that can do temperature and something that can do the heart. A lot of devices just have heart rate, for example, and so they will miss this upward trajectory. Similarly, if you just had temperature, you'd miss this dimension here, right? So the combination of different modalities and how we test that is a big issue in the field right now. There's also a lot of people right here who report, in this case, very mild fevers, and we can't tell that apart from daily noise. There's enough change day to day amongst people that you don't want to be sending a lot of false alarms to somebody every day that they worked out a little extra hard or walked extra fast to get to the bus. Their temperature goes up and you go, hey, you're getting sick. People will burn out on that. And so this is a really interesting challenge to say, how far ahead of something do you need to get? What are your false positive and false negative trade-offs in terms of both surveillance and, again, feedback to the individual thinking of behavioral modification? It's nice to celebrate the wins. And of course, we do this in the literature, saying, hey, this thing is possible. We've published a new version of a new fever classifier. It's just also extremely important, I think, to temper that excitement with the challenges of how complex human life is as a signal against which you're differentiating these things. So again, we can zoom in on this and we can start to lay out territories of true positives doing very well, looking very segregated in this same space. So what we've done here, it's the same data, it's the same map. We've turned it into your sort of hiking topography, topological map. So here's this sort of peak of true positives, but they carry this large drainage basin, if you like, fairly separate from true negatives. But then there's these false positive, false negatives overlapping in the middle, right? And so again, more work is needed to figure out how do we make this more robust and for whom are we making it robust? That brings me to this. When we looked at our COVID analyses in the wild, so this was not that same fever classifier, but rather classifiers built on those 6 graphs of COVID data that I showed you. One thing, again, we can revisit, right? How important are these different modalities? So how much does performance decrease when you remove skin temperature, heart rate variability, respiration rate, activity? And so we can start to weigh, on average, how much was each of these features or each of these modalities contributing to this algorithm. Again, because a lot of off-the-shelf wearable devices have different sensors in them, I think it's a really nice thing to push for standards to say when we're going to use these in the wild, when we're going to actually affect people's lives. Let's make sure we know the trade-offs of these different sensor choices of these different data types. And there are systems for doing this. The other thing, much harder, is to say, well, for whom is this working? So a lot of algorithms tend to look for a single global maximum, which is to say, this works on average the most, and it's the single one-size-fits-all solution for the market space. I'm not a fan of that because people are different, as we've already seen. So this is an illustration, again, published in our work on COVID. We're very happy that we were able to show pretty quickly that you could do wearable device detection for COVID. But we also wanted to highlight the shortfalls. So here we're seeing that the algorithm works on average 7 or 8% better on men than on women. Digging into this further, what we found was that we could develop techniques that identified women who were cycling and non-cycling. In this case, now we're talking about monthly rhythms instead of daily rhythms, but you can imagine it's a similar principle. And on those women, it was doing a bad job because when they ovulate, as I've said, their heart rate goes up, their temperature goes up, it goes, oh, you're getting sick, and it says, go get your COVID test. Because those things are not exactly stable, women are not 28-day metronomes, you need, again, customized, individualized baselines with some probability estimates in them. We can build those, we can bring these scores back up into equity with the male scores, which is really nice. But it requires your awareness that the populations you're serving are different and have these different physiological dynamics. So going forward, that's going to be one of the major challenges in the field. It's not just saying, okay, men and women exist as a binary. It's obviously much more subtle than that. Some women cycle, some women don't. Some women are more stable, some women aren't. Men and women both experience the weekend differently or the day differently. And there ends up being quite a lot of complexity to the timescales that make one person's data similar to themselves over time, similar to, let's call it, their neighbors over time, different from other people. So this development of augmentation of things like demographics, male, female, age, race, et cetera, we want to add in data-driven sort of new demographics, which we might call physiotypes or something of that nature. This is very important. I've said that I study women's health. I want to just remind everybody, work from one of my mentors at Berkeley, Irving Zucker, and Anand Spiri, who's now a professor there, going back through reviews of very large numbers of articles, find that women tend to still be excluded from research as subjects. So what we're looking at here is different fields that from one of these reviews they've published, and there's unfortunately, it's an ongoing series of these reviews because things are not actually changing rapidly. But in animals over here or in humans over here, what we see is that if there's only one gender reported, it's usually males, shown in red. Sometimes both genders are included. That's of course, that's a good thing, but it's not often an even mix. It's simply, yes, some females were included usually. The only field where females tend to dominate is reproductive physiology. And this reflects, an unfortunate bias in the framework of how experiments are carried out, that, women are different because they ovulate or get pregnant or something like that. And that other aspects of health, basically should be the same as male. And of course, by now we know that's not true. And so reminding people to try to have this inclusion, but also to try to analyze these groups independently. And I think in the future, map out what are those dynamic physiotypes that really identify people as similar or different with better resolution than just sex, should be very helpful in overcoming this burden. We've published a number of analyses now that try to numerically look at, is it true that males and females are in fact statistically more variable than each other? And it is true, the males tend to be more random than the females. Females tend to be more structured in time. So we've probably been looking at this backwards, if anything, but I think the pragmatic solution is not exclude anybody because it makes the statistics hard, but rather include everybody, statistically analyze them appropriately, and let's make sure the science is serving everybody. One way we can do that is, again, IoT, right? So this is now to say a lot of these studies are done because statistical power is something people are worried about. That's only the case when your data are very small, right? So in hospitals in the US every year, we have about 2.5 measurements per person, basically, 7 billion tests over 3.5 million people, or 300 million people, depending on how you slice it. Wearable devices, at a conservative estimate, are generating tens of thousands of times more data than hospitals are per person. And that kind of data availability, without looking at your Facebook posts or your email frequency or your Twitter post timestamps or your learning management system school stuff, just the wearable devices alone is clearly going to grow faster than the number of hospitals will grow. So the data in the real world vastly outstrips what we have in the sort of standard clinical framework. That's a challenge because these are not clinical standards. But I think that it's kind of crazy not to try to figure out where is the information within this vast, sudden abundance of data that lets us do a better job of just capturing naturalistically. Okay, what do men look like? What do women look like? What do old people and young people look like? How can we get past these very broad categories into much more data-driven, nuanced categories? So I'm going to pivot here away from wearables for a minute so that I can talk about some of these ambient data, some of these data that we just generate anyway. Here we can start to, I'm sorry, this is me continuing physiotypes. Let me just round that out. Again, the idea that there are women who are non-cycling, this is temperature across days, this looks kind of like noise. These are 10 males, 10 females. They're pretty indistinguishable. But hey, here's 10 females that we can align by cycle time, and we can see, okay, they're warm, they're cool, they're warm, they're cool. So just saying males, females really doesn't capture this, right? We need these physiotypes to capture what kind of male, female, what physiological dynamic are we interested in? Okay, so ambient data. These are data that show a nice daily rhythm. They show a difference between males and females here. Males tend to stay up a little bit later at night. They tend to be a little bit less similar to other males, and so that average stays higher. It never dips as well. Shows age differences, shows seasonal differences, and I'll expand on this in a second, but these data don't come from wearable devices or any effort at all by these individuals. These were scraped out of the campus learning management system. And so when an individual logs into Canvas, for example, this was actually done at Northeastern Illinois University, but it's the same thing as Canvas, they generate a timestamp. And people tend to generate timestamps that are roughly consistent over time. And if you look on days of classes, as I'll show you shortly, it's very masked by that social demand of classes. If you look on the days when they're not registered for classes, they actually reveal a pretty good individual biological signature. And so when you watch somebody across a semester, say, you can start to build these maps very reliably. Okay, this person's here, men are more active here, women are more active here. For age, here we've colored it by decade of life. NEIU is the most diverse institute of learning in the Midwest, and it is community serving in the Chicago area, and it has this very broad capture of ages, all the way up into the 80s and 90s. Here we've just done decades of life, teenagers, 20s, 30s, 40s, 50s. And the fact that this makes this nice rainbow here, helps us see that just like we'd expect, older people are getting up earlier, and these data reflect that with pretty good fidelity. Similarly, things that are hard to study and don't have standards emerge. So here at the bottom, we're looking at seasonality. We're looking at the difference between fall and spring semester. So in the fall, days are getting shorter, and we see that there's this consolidation of activity towards the middle of the day. Whereas in spring, in red, days are getting longer, and we see that activity tends to start to spread out into the evening and the morning, consistent with this idea that human behavior is modulated by these natural rhythms. So as I said, we can take this not just at the level of the population, but all the way down to the individual. And we can start to rank people based on the phase of when are they most active, what we would call the chronotype. So here you can notice most people are very inactive between midnight and 6 a.m. They're asleep, presumably, right? I don't know them. I don't know anything about them. but just from their activity, I can guess that they're probably asleep here, and that's consistent with our expectation. And then you can see there's some morning types, there's some midday types, there's some night owls maybe. And so even though these are fairly low, coarse data, low grain, even though I don't really know a lot about these people, because there's so much data, because it already exists, with no funding, without any great effort, we can start to build these personal predictive models of when is this person most likely to log into their class system. We can then go back to that jet lag where we started, and we can say, so here's Aaron Shermer, my friend and collaborator at NEIU. I showed you jet lag on an individual initially, and of course, I think you all know jet lag feels unpleasant, right? None of us enjoy that. There's good literature to believe that shift work, which is just sort of prolonged social jet lag, is also really bad for most aspects of health: cancer, osteoporosis, early onset of dementia, all kinds of things. So we might want to know, is jet lag happening in the real world? Is it happening to lots of people? Amongst those lots of people, to whom? And when we see it, does it really correlate with negative outcomes? So these learning management data give us one example of that. So here again, people are mostly asleep 0 to 6. We see this range of chronotypes. Here I've made up some little bird cartoons. So you're a morning lark, you're a daytime finch, or you're a night owl. And we can compare that in the same students in the same order to the day of classes. So again, this is days when they're registered for class, or not registered for classes on top, and when they are registered for classes on the bottom. Same students, same order. You can't tell who's a lark or a finch or an owl. Everybody's just on class time. This is a social imposition on top of the biological rhythm shaping that activity. What that means is that for every individual, we can calculate that phase shift from their row on the top to their row on the bottom, and we can give them a number. How socially jet lagged are they on average? We know that feels bad. We know that's probably not good for you. Does it affect something in the real world? Well, these students generate grades. Those are a metric of cognitive performance. Does social jet lag correlate with cognitive performance? Yeah, very strongly it does. And so here we see people that are getting up earlier than their class days, and even they have some imposition. Somewhat expectedly, the later that people would like to get up, but then are made to get up earlier for class days, the worse they tend to do in those classes. We can break this down further. We might say, well, gosh, maybe it's just because the night owl is taking a morning class. I bet they do fine in their evening classes. Unfortunately, as I showed you in that first slide, jet lag persists for a while. And so if you're jet lagged, say you have Tuesday, Thursday classes, you're jet lagged 2 days every week, you may never get back to stable. And consistent with that, what we find is that all of the students do worse in morning classes. So here we have time of class, type of person, and then little cartoons for those things. And the stars are significant differences. And what we see is that everybody does worse in the morning. The later classes, everyone does better. And yet, owls always are doing worse. And so from a public health point of view, if you don't know that chronotype is a demographic, you don't know how to try to capture these effects and find that there's this population that's being negatively impacted by, in this case, our choice of school programming. When we can get these data, when we can look for these biological patterns, we can identify these people pretty readily. It's worth recognizing that chronotype and circadian rhythms, a lot of sleep for that matter, have pretty strong genetic components. And so, I tend to think that when there's a group of people with a genetically distinct identity whom society disproportionately disadvantages, normally we would call that discrimination, right? If this was skin color, for example, you'd say, okay, well, wow, something's really wrong. There's this population that's being disadvantaged systematically. Same thing's happening here for these biological patterns. We haven't been able to see them easily in the past. Now that we have all of this IoT data, we can surface these patterns. We can see them. That's really important if we're going to be able to intelligently address their needs and make sure everybody is served equitably. Coming back to sex then from that, we also were looking at, even without wearable devices, how well can we do at COVID detection? Less well, but pretty well, actually. So a very talented undergraduate student who's just graduating Amit Klein, developed a Bayesian network model of symptom reports. So I told you we had tens of thousands of people's wearable data. They also filled out several million survey responses for us over the year. So what we did was we said, okay, well, let's look at the frequency that they're reporting symptoms, and let's see whether men and women are reporting that the same way. This was inspired by work on heart attacks, where a lot of women were being sent home because instead of saying, hey, my left arm is numb, they would say something like, well, I feel queasy or I feel nauseous, because heart attacks manifest differently in men and women, different symptoms. And so, then they go home and they die of their heart attack. It wasn't until the 2000s that this was starting to be recognized and addressed. So we said, okay, well, COVID's a big deal. Let's make sure we know what's happening here. So we ranked the symptoms by the frequency that they occur in females in blue. And of course, what you can see is that red does not share that ordering, right? So the males are different. We can also look across time by the frequency of symptoms. So same symptoms, now we're looking at their relative abundance by days leading up to the day of diagnosis, day 0 here. And this sort of orange square that you see is highlighting that two things. One, men tend to respond and give you symptom information much less. So just as a patient population, there are behavioral differences. Men are less good at reporting symptoms, hence so much blue. But also when they do report them, they report them differently, right? They're reporting more aches and pains, they're reporting more fatigue, and they're reporting more fevers. So then we were able to develop this into rankings of screening questions, working with clinicians at UCSF to say, if somebody comes in and you believe they have COVID, you might ask different questions about what are they experiencing to be more accurate across sexes. So what does all this lead us to? And this is where I'll end my talk. The point is to say, when we started COVID, we had tools like this coming from UC Davis, trying to share in as close to real time as possible case report loads. The challenge here is that getting a case, logged in the hospital, sent to the county, eventually, hopefully sent to the CDC. At the start of COVID, this was about a 4-week loop. The dream would be to say, if we have individuals generating data, if we've solved access so that we know that different neighborhoods are getting information equitably, if we've solved security and privacy so that people are opting in and want to be sharing their data to be part of syndromic surveillance instead of, feeling creeped on or big brothery, we could have individuals acting as tracer particles across the country, across the world, and we could see that day. Here's this person, these are real data. Here's this person, hey, they're getting a fever. Let's go see what's going on, rather than waiting for weeks to pass to know that they had gotten a fever weeks in the past and that something was going on, at this time when I took this picture off the internet, Arizona and California weren't doing so well. We could see these people and close that loop much faster. The science is totally ready for that. It's really a challenge of taking the engineering science, integrating it to public health best practices, which is why I'm reaching out and hope to be able to collaborate to support these efforts, because the potential is to have weather radar for health in a way that we just don't have yet. UCSD is the place to do this. We have so many wonderful centers here that are well-resourced, full of incredibly collaborative experts. Institute for Engineering and Medicine has funded me. I'm a member of Center for Wearable Sensors. Of course, Rob Knight's Microbiome Center is what's given us all of the sewage testing that we've been getting over the last few years. School of Medicine, School of Engineering, Hillsborough Data Science Institute, the new School of Public Health, which is extremely exciting to me. And then a lot of this work is supported through collaborations with the Supercomputer Center. So, what are the right data sources? How do we make sure that they're capturing diversity in the population? How do we do that in a pro-social way? How do we make that, a stable reporting system that's trustworthy? And how do we bring that back to the individuals that are generating these data to help change behavior? I think these are the challenges that are in front of us, but addressable. And I find that extremely motivating. So I will just end there. I'll say thank you to my lab, TempPredict for providing funds, which is UCS clinical led that I've been the technical lead on. Aura Ring providing access to those data for users that opted in, Quantified Self, Department of Defense, and MTech, which provided the funding, and of course my home institutions. And with that, I think I will stop sharing, and I've got my little animated extras. I'll stop sharing, and I'm happy to take questions. And Steele, thank you for inviting me. I really appreciate it.
Oh, Ben, my pleasure. It's a wonderful talk. I thought it was very interesting. As far as syndromic surveillance goes, it sounds like it's still a future goal. Is anyone actively working on that here at UC San Diego or no?
Yeah, I mean, we are, Topol's group is, I believe there's a few others. In fact, Chris Longhurst was just given a to try to create a sort of a control center at the hospital, and that involves this conceptually. How we actually implement that, yeah, absolutely. Those are ambitions we need to realize. That's exactly right.
That's interesting. I mean, years ago when I was in grad school, one of the doctors here, ID doctors, wanted me to set up a county-wide syndromic surveillance system. And that, I thought that was very daunting, of course, and that we're talking a long time ago, and here we are. It's still something happening, tomorrow or the day after. It's extremely important that we get that information, and now that we have the data sources, right? So I'm curious too, as far as that getting the buy-in from people, are there any strategies or thoughts on how best to do that so people aren't creeped out, as you say?
I think it's very cultural, and I think this is where one of the reasons I like to give talks like this is finding good partners, right? So, I'm a 6'2", blue-eyed, blonde, white guy. I fit in some groups, and I have no connection realistically to others, right?
Right.
I grew up in the Midwest. I have some connection to that culture, but the sad version of the story is we had a lot of people donate data. Again, 100,000 people reached out to us willing to share data. Huge bias towards white, not a huge bias in sex, but male dominant, and huge biases towards things like California or Western Europe. And meanwhile, at the same time, down in Imperial County, at the border here, the COVID rates were 10x what they were in La Jolla, right? As hugely more burdensome. And we even wrote a Smart and Connected NSF grant to try to reach out. And it was just nothing we were able to get traction with, right? What we need is embedded community partners that have trust, have earned trust within those communities so that we can even start to have those conversations. The feedback we got was, you're the 50th person from UCSD to think you're cool by saying, hey, come tell us your story. We go and tell a story. We never get anything from it. So, screw off, right? Totally understandable that there shouldn't be that kind of a relationship. So how do we do a better job of building trust is a big part of this. One of the ways I've tried to address that is I'm building collaborations with minority serving institutions. So NEIU is an example of that. They're deeply embedded in Chicago. They not only have much larger representation of Hispanic and African American, but they have, unbelievable students who are war refugees from Syria and have come over here and are trying to get their education. And I mean, just incredible, enduring people. Getting to meet them, getting to work with that university, saying, how can resources from UCSD strengthen, for example, data science training so that you can bring that back to your communities? I think that's one of these opportunities.
That's great. I mean, and then, we're all working so hard to find the solution, but as you said, it's going to be more localized, or, will be at the most maybe a trial in the county, but then we have to build the tapestry county by county to the country. So it's just a huge undertaking, extremely important. I was also interested, as far as chronotype goes, you did say there's a genetic component, right? I personally am a night owl, right? So I'm just like, we can try to alter on a conscious level our sleeping pattern, but it sounds like you can't get away from the way you were made, basically.
The evidence suggests that that's right. So, one version is, for example, if I fly to France, eventually I get onto France time. But if I'm an early type here, I'm probably going to end up an early type in France. If you're on rotating shifts, like I said, you have all this social jet lag, it's worse for every part of your health.
Right.
But if you're on a stable shift, I just always work the midnight shift, let's say, right? I'm not as bad as the version of me that's on the rotating shifts, but I'm worse off than the version of me that isn't on the midnight shift. So you don't just become that new stable time.
Okay. That's great. Anyone else, any questions? Please feel free. Gene, I'm going to put you on the spot. Oh, Sanjay has a question in the chat. So let's,
Is
Ben, did you see
chronotype
that?
associated with personality? That's super interesting.
Hmm.
And it's a little bit circular, right? So I want to make sure that I would be very clear to say causality is very challenging in this case because of the fact that, for example, we know that late types have more trouble in school even as young kids, hence the push to delay school start times, which CCB has had quite a bit of success in. Proud of that. The problem compounds, right? So you say, okay, well, kid wants to get up later. I'm not letting them. They're kind of cranky. The teachers give them a hard time. They don't put them in the advanced placement class. They're feeling a little disenfranchised by the time they get to high school. These things snowball, right? And so it's reasonable to assume that if your brain is growing up with a different alignment to the world, it probably reinforces different traits. It's reasonable to assume, the old joke is all the night owls that end up in computer science and theater, I guess, right? But of course, is that real? Is that biological fate? I don't really believe in biological fate in that way. But are there predispositions that are reinforced by that interaction with the environment? Probably. Again, I think the question is, given that complex landscape, historically, you had to say, well, look, everybody just needs to synchronize their mechanical watches because how else will they know what time it is, get to work at 9? We don't live in that world anymore. We have enormous amounts of customizability. We've been doing remote learning and remote talks like this for years now. How do we take advantage of that instead of just complain that it's not standard, right? How do we do a better fit to the much more complex human landscape?
And so chronotypes, I mean, as far as this has really become more of a thing because there's more available data, right? It's the Internet of Things. So like say 10 years ago, there were a few people discussing it, but now it's much more prevalent. Is that correct?
That's exactly right. So, and in fact,
Okay.
I was one of those people. I used to tape things to my body and demonstrate that I could see these rhythms in different conditions and, give myself ulcers from, or, sores from wearing a device too long. Can't recommend you try it at home. But then cell phones became smartphones and wearable devices became a market and schools started implementing learning management systems sort of as a standard. So yeah, it's absolutely been just sort of 10 years of this explosion of possible data. I work with Intel, for example, and they have huge amounts of data on just device diagnostics, nothing personal at all. And so, I've been working with them to say, yeah, but a human drove that. I bet there's rhythms. I bet that lets us see, for example, which regions were more hit by COVID, and it does, right? Sure enough, they line up. So a lot of people have these data, and I think medical systems is an example, education systems is an example. The question is, what do you do with that data, right? Are you really juicing it for all it's worth? And we're not coming close to that. We've accumulated it much faster than we've accumulated the knowledge of what to do with it.
Well, at least it's some job assurance for you.
It
There's
is.
always
It
plenty
is.
of work, more analyses to do. Yeah.
I don't have to be, thank God, I don't have to be that smart. I just have to be the first guy to kick over this rock. That's right.
Oh, wow, that's great. Anyone else have any more other questions? Please feel free. Gina, I was going to
Yeah.
put you on the spot, but don't worry about it if you
Yeah,
don't
no,
have
no,
one.
I do have.
Okay,
This is a
go
really
for it.
interesting talk. So have you looked at like any chronic diseases that you could kind of compare to, like like what you mentioned, long COVID, right?
Mhm.
It has very similar, I would say, symptomology to MECFS types of
And you'll have
behavior.
to break that down for me. I'm sorry. I just play a clinician on TV. What is
So,
MECFS?
MECFS is it's like chronic fatigue syndrome.
Okay, got it.
So like they have very similar symptomology,
Some of
and
them do. Right.
some
It
of them,
depends
yeah,
where
yeah,
COVID damage occurred, but yeah, absolutely.
right. And that's the problem with, like, even within that that symptomology, that you
Yeah.
really can't put a finger on it, whether or not it's a viral, is it a viral etiology, is it autoimmune etiology, or a bacterial. And have you, are you planning on doing some really interesting, comparisons between, that type of
Yeah.
disease and long COVID and even acute, and whether you could predict whether someone would become long COVID, will develop long COVID?
That I would love to do. The ultimate hope of this sort of physiotyping vision is that you would, in the grossest terms, you'd sort of separate better expected responders and non-responders for some intervention, right?
Right. Yeah, exactly.
And so I think that's certainly on the table, right? Can we do better than your male and female, or you're old and young?
Right.
Yeah, no question, right? No question. Steele and I are not physiologically identical, however similar we might look on Zoom, right? And so, and whether or not I've been keeping up on my exercise or not is sort of local to me in time.
Right.
We are looking at some long-term things, right? We're getting into mental health. We're getting into chronic inflammation diseases. One of the things that really excites me about this is there's the landscape of people, right? What's physiotype? Who am I versus who is Steele, for example? But then there's the landscape of manifestations, right? Let's say that he and I are very similar and we both get COVID, but we don't get the same manifestation, or we both get depression, but we don't have it in the same way.
Sure. Yeah.
These things are kind of like syndromic labels, are very much like demographic labels in my mind, right? Depression is not a thing. Depression is a very complex landscape itself. So can these data help us do a better job of saying, oh, okay, you're depressed and you have this cyclic quality to your sleep fracturing, and we can see that, and it's coupled to inflammation in your temperature, and we can see that suggests a different treatment than these other groups, right? We're not there.
Yeah.
But the ability to do that is now on the table, right? So that's what we're trying to crack into.
Right. So the data that you've been showing, is it like an aggregate from a lot of different devices, or do you
Yes, and the data in this talk was mostly Aura data. I showed you some Freestyle Libre continuous glucose data. I showed you some other temperature probes.
right.
The lab works with whatever it can get its hands on. So we have this data from TempPredict, which was Aura data. We have about 10,000 Dexcom user data. We have millions of Intel device data. We have a sort of growing library of interesting sources of data. And then that's really nice, because one thing I didn't get to talk about at length is when you make an algorithm with sort of a global maximum, you don't necessarily know if that global maximum is just specific to that data or even that data structure, the database, if you like, as opposed to actually it's a physiological thing, right? So, for example, convincing people that, I see the daily rhythms and I expected that, and of course I'm going to see it in heart rate and temperature and online activity and whatever else, until you've seen that that's really in all these different data types, it's hard to say, okay, daily rhythms is a thing. It's not just daily rhythms of online activity, for example.
Right.
And the more subtle patterns, obviously are less well accepted than daily rhythms. So that's the challenge, right? How do we know these things are really going to generalize and be useful to other populations?
Right.
That's great.
That's really
cool. Oh, go ahead, Gene.
No, I think it was a really fantastic talk. So really
cool. I appreciate it.
Yeah.
And Sanjay has his hand up, and I think we'll wrap after that. So
I gotta go, but
yeah.
Thanks, Gene.
Thanks, Gene.
Great talk, super interesting.
Cool.
So we tried to do a syndromic surveillance project a little while ago in San Diego, and trying to get schools to ask maybe 2 more questions other than, when someone calls in to say my kid is sick, give me 2 more symptoms, and then use that. Is it a GI, respiratory illness? But we couldn't get people to do it. And it kind of boiled down to is like, there's a threshold amount of work that's needed for people to share data, and wearable seems like that takes it away, right? So that's not the issue. So the issue has to be privacy concerns must be the biggest thing. And I'm just wondering if what sorts of creative ways have you guys thought about to kind of alleviate those concerns? Because nothing needs to be individual level, right? Like
I don't know. So sorry. I think you asked a bunch of questions actually, but maybe you can email me and we can get coffee sometime and talk it out. That would be fun. I think getting the information back to the individual is actually a big part of the payoff, right? So a lot of the compliance that we've had success with is individuals say, okay, I care about COVID, I'm willing to donate my data, but I want to get an alert if I'm getting sick, if I'm going to be part of this study, right? And so rather than say, can you please take on an extra burden for society, we try to say, can you sort of be selfish about this, but share that selfishness, right? In the way that I might share pictures of what I'm eating, and that gives rise to food blogs as a thing, right? Or I might review a restaurant that I liked, but then that affects whether you want to go to that restaurant or not. to the extent that we can get people to do that, that's great. There are still lots of issues of self-selection, right? People had to want to get a device. They had to want to be involved. They had to feel important enough, or that their voice counted enough to believe they should be part of a survey. Those are pretty culturally specific assumptions that we can't just make for everybody. And so, again, I think there's a lot of work to do to say, what are the right sort of cultural, educational efforts that we can make to bolster these things, so that it's not just, enabled, enfranchised, confident people like me that go, of course, somebody wants to hear about my symptoms. How do we get that into a broader world and make sure those people are, the people that are less vocal are nevertheless getting served?
Yeah, that makes sense. I think it's funny, I think about myself in this situation, and there's definitely an age gap. I don't use Facebook. I don't share anything at all. And,
I mean, I don't use Facebook. I don't do social media either. It's
the kids I know, my son and his friends will share anything. Yeah.
I think that's, weirdly, I think that's kind of an opportunity. And I'm sorry, I know we're over time. I'm late for my faculty meeting, but that's all right. This is fun. The generational thing is real, right? So it's definitely the case that younger people are more likely to say, "You're stealing it anyway. Why would I care? Have it." I think the opportunity again is to say, okay, but how do we get away from just sort of people who already use Facebook and are cynical about privacy to, people with, again, thinking of the border populations, very legitimate personal and family security issues that stop them from just wanting to share their location, for example, right? How do we make sure that sort of like when somebody has a drug problem and comes into the doctor's office, that doesn't just mean they're going to go to jail and so they don't seek treatment, right? We need to find ways to do this where the data are segregated so that they are for public health service and not, ratting people out one way or another for something someone else wants. That's not a science challenge. That's not my field, but it's clearly needed to get this into the world.
Yeah, I think, I know we're running over too, but I think there may be a science problem in there as well.
Well, let's talk about it. I want to make sure I respect everyone else's time as well as the faculty that I'm coming in late for. But
well, I definitely think you two should chat offline. Yeah,
exactly.
Yeah.
Please reach out.
Just a few closing words, and Ben, thank you so much. I know you thanked me, but no, this was fascinating, and it really just opened my mind to the possibilities and all the work that there is to do. I mean, it's just amazing, and I would love to have you back. Should you have any more developments in research, or there's movements in the field, that'd be wonderful. So, and then as far as the talks go, they're every month. We don't actually have a speaker lined up for next month, but I'll get to that shortly, and then we'll send those details out. But please do participate in these, attend them when you can. It's super important, and it's a great public profile for the institute, and we're very grateful for you, Ben, and your time today. So I won't hold anyone else up yet. Fascinating talk. Great. I wish everyone a great day, and take care.
Thanks so much.
Bye-bye.
Take care.
All right. Cheers. Bye-bye.